Hi everybody. Today, are you having another headache with poor Western results? Western blot is a protein analysis technique commonly used in biochemistry, molecular biology, and genetic immunology. Anyone who has done Western knows that Western is not really a good experiment to do. It is just like a shot in the dark. So next, we'll be updating a series of videos. We will analyze the possible causes of some abnormal Western results and solutions. So in today's installment, we'll take a look at what happens when we can't get the bands to come out of the exposure or when the bands are weak. When we see this, usually we think of extending the exposure time. But that's not the only solution. When longer exposure times still don't do the trick, we're definitely going to check Western's entire process to figure out why. If the problem is with the sampling, what are the reasons that lead to this result? Well, the first reason is that our target protein is not expressed, or the signal of expression is long. This requires that we must refer to the literature or consult UniProt before doing Western. Go and check the expression samples, expression level, expression molecular weight size of our target proteins, and whether certain stimulus conditions are required to induce its expression. Also, when we extract the target protein, be sure to select the appropriate lysate based on the localization of our target protein. Let's say, I want to extract nucleoproteins. I'll take the nucleoprotein extraction kit. If I want to extract mitochondrial proteins, I'll just isolate the mitochondria first. The mitochondria are then cleaved with a mitochondrial lysis solution and get mitochondrial proteins. In this way, a maximum yield of the target protein can be achieved, and enrichment of target proteins at the same time. Second possibility, the samples are degraded. Degradation of the sample will occur during lysis. Then this requires that the sample from which the protein is extracted be as fresh as possible. We must keep the temperature low during extraction. Before extraction, be sure to add the appropriate protease inhibitor to the lysate. If it's a phosphorylated protein, phosphatase inhibitors should also be added. Then after the extraction and measurement, at the loading buffer, samples are heated in loading buffer for 3 to 5 minutes at 100 degrees Celsius to aid in the denaturation. After finish that, storage in separate packages. When use the samples, always try to avoid multiple repeated freezing and thawing of samples to minimize sample degradation to the greatest extent possible. Third possibility, our lysate is too inefficient or not lysed enough. Then it requires us to choose the right lysis fluid based on the localization of our target protein. Just like what I said, if you want to get the nucleus protein, select the appropriate nucleoprotein extraction kit. If you want to get cell membrane proteins, please select the appropriate cell membrane protein extraction kit. This makes your maximum yield of target proteins for enrichment of target proteins. For an adequate protein cleavage, be sure to allow enough time for cracking during the cracking process. Then for conventional REPA lysate or Western and IP lysate, typically it's 10 to 15 minutes or less to crack it. During the cracking process, for cell samples, be sure to turn it upside down and mix it up from time to time or blow occasionally with a pipette. This ensures that the lysate is well mixed with the cell samples to achieve the effect of sufficient cracking. For cellular tissue samples, please always use physical crushing to achieve full protein release. Fourth possibility, the target protein isn't much enough. If it's a regular protein, the total protein is generally 20 to 40 micrograms. But if it's purified protein, the total protein is 10 to 100 nanograms.